Hello students, so we had a extensive discussion on the formalism of UV visible spectroscopy, basic concepts also we discussed, and many of the application we also discussed. Now slowly I want to give you some idea how we can use in high end biological research as well as say industrial research. So in few lectures which is coming from today onwards, the course will be touching upon the usage of UV visible spectroscopy in advanced research or in a biopharma or diagnostic research and all those. So today we will be just touch upon the concept that we had already discussed, hyperchromicity and hypochromicity, and we will build upon that. So what we are going to discuss is the difference. You remember we had already discussed hypochromicity and hyperchromicity, right. So double stranded DNA absorbs the less UV light because of the base stacking, and when it unstacks, the single stranded DNA absorbs about 30 to 40% of more base. That is a hypo versus hyperchromicity. And this basically happens because the DNA is melting, right, the double stranded is melting, and because of this heat that we are putting it, the heat is breaking the hydrogen bond, it is unstacking the bases, and therefore absorbance increases as the strands, two strands which was twined are coming separately. So using these we can get the melting curve of the DNA, we can get the TM, the transition midpoint of the curve, and this looks like a sigmoidal curve. We will be seeing it at the 50% of denaturation, we can calculate the TM, and that essentially if you do these experiments, that essentially reflects the DNA stability. So we have also touched upon this concept that if GC content is more, that means TM will be more. And so therefore, it not only tells the stability, but also it tells about what is the sequence, how long is our DNA. So length of the DNA can also be understood from that. And then we can calculate some of the thermodynamic parameter like a Van Hoff analysis, where we can derive the enthalpy, entropy, the free energy of melting curves. Subsequently, we are going to touch upon what is the effect of different kind of solvents and salt like a denaturants, urea or formamide, which can lower the temperature, the salt, right, what is the effect of salt or pH, all those, and how we can use this concept in quantitative PCR and HRM methods, and how we can use this melt curve analysis to detect what kind of mutation, whether it is a stabilizing mutation or destabilizing mutation, which can distinguish the wild type from the mutant, and this can enable us as a diagnostic tool. So we can use simple concept of this melting in coming up with a diagnostic detection of the DNA. So just to catch up what we had already discussed between the difference between hypochromicity and hyperchromicity. So hypo, decrease in the UV absorbance, wherever there is a decrease it is a hypo, where there is an increase, that is hyper. This hyper happens single stranded, hypo happens the double stranded, because your base stacking are there and now base are separated here. So this is the characteristic of a double strand, this is the characteristic of single strand or partially melted DNA. Now what is the cause or mechanism? In double stranded, the base are stacking, the hydrogen bond is forming, and that restricts the electronic transition, that reduces the light absorption. Whereas in single strand the loss of base stacking happens, the hydrogen bonds are not there, and then that enhances the electronic transition, and that is why there is a increased light absorption, and therefore you have a more intensity. Now, so because of all the stacking hydrogen bond, the hypochromicity, the absorbance is lower, here absorbance is higher. The aromatic base are tightly stacked, right. So, therefore, UV excitation is lower, here it is unstacked, more exposed, allowing maximum UV radiation. The typical experimental condition, right. So, this happens at so occurs under low temperature, the neutral pH and stable ionic environment. But if you increase the temperature or change the pH, basically it will unstack, the hydrogen bond will be broken and you form the single stranded and therefore hyperchromicity. Now, this indicates the stability, this indicates the instability of DNA, and we can use this hypochromicity for duplex formation or renaturation, and if it is unstacking, the high Tm will tell you that it is single stranded. So, one can use in denaturation, renaturation study. And now, this is example like a native genomic DNA or fully annealed polymerase chain reaction product, PCR product, and here if you heat the same DNA, the Tm will go up. The first cycle of PCR denaturation, you can see the hyperchromicity. So, let us expand upon it. So, what we are saying, like it is going, so if you are putting heat in, this is the cold DNA, that are double stranded. If you are putting heat, it is going to hot DNA, and you can see the base pairing is breaking, the unstacking happening and DNA is splitting. That is the hyperchromicity, high absorbance, right. So, if you are taking a double stranded DNA, slowly you heat, what happens? Denaturation happens. Denaturation goes by a progressively, it goes for cooperative transition, that we are going to see sigmoidal curve happens, and then it happens where the DNA melts in a narrow temperature range. This transition appears the smooth melting curve, which can be monitored by simple UV absorbance. Right. So, Tm is a temperature at which 50% of DNA remains in double stranded and 50% becomes single stranded, right. So, this represents the thermal midpoint of denaturation curve, and this is one of the key parameters that is used to determine what is the what is the midpoint of the thermal denaturation. So, where 50% is in double stranded, 50% is in single stranded, that is the Tm, right. So, if you look at here, this double stranded DNA absorbing at 260 and we are measuring absorbance at 260, and slowly in a narrow temperature range, if you look at, it just moves to high absorbance. Now, it becomes single stranded DNA. So, on x axis we have temperature, y axis we have absorbance as 260 and the midpoint of that is called DTM. So, principle is extremely simple, as it denatures, the absorbance increases. So, double stranded exhibit lower UV absorbance, hypochromicity, and effect of heating, hydrogen bonding is broken, base stacking interaction gone, and therefore, it absorbs high, and you can find it out what is the midpoint of temperature, right. So, that is what happened. Denaturation produces distinct increase in the UV absorbance. The single stranded DNA absorbs strongly, this one strongly absorbs, and the change in absorbance provide a reliable and real time measurement of DNA melting behavior. So, looking at this curve, you can find it out, the Tm and whether this DNA is in single strand or double strand, and what is the percentage of single strand and double strand, that also you can find it out. So, that is the hypochromicity, this is the hyperchromicity. So, melting behavior is a sigmoidal, here you have a baseline, here you have a last baseline, and then you like when plateau is achieved, you know that it is completely denatured. Here plateau is like if you start from renaturation, when the plateau is achieved here, you know that completely double stranded DNA, right. So, that is a 50% melting. So, correspond to midpoint of the melting curve, and this is the key parameter that determines the DNA stability, also content of the DNA, whether it is AT rich or GC rich, because GC, you remember, there are 3 bonds, right. So, breaking 3 bonds require more energy than breaking 2 bonds, which is in AT. So, if melting Tm shifted in this direction, that means it requires more energy and therefore, it is more stable, right. So, one can get the content of the bases that are present in the DNA, and this is also used for optimization the PCR or hybridization, and also can be used such concept in molecular diagnostics, right. So, now what are the parameters that determines or we can determine using UV melting curve. So, as I mentioned, the Tm is extremely important, right. So, Tm is extremely important, that extremely important parameter that also tells about the GC content. So, let us do some experiment and find it out what we can get. So, let us do these experiments where we are having two single strand DNA with a complementary bases and we mix it, right. So, as soon as you mix it hybridizes and forms a duplex, because the bases are complementary, right. There is no mismatch, it is a complementary bases, and when you increase the temperature, when you are increasing the temperature, it goes from hypo to hyper, right. So, that is what it happens, and now you can basically you can look at this is the curve. So, if this is the curve, lower concentration you are increasing the concentration and increasing the concentration and increasing the concentration. So, if you increase, if you look at absorbance is increasing, right. So, absorbance and also you can notice little bit maybe it is shifting little bit. So, essentially it says that when the concentration is more, right, absorbance that it starts is more. Here if the DNA is of same type, you can see it not shift much, but if DNA content changes, the Tm the Tm will shift, right. So, looking at the fraction of association, we can find it out how much is in which condition, whether it is duplex or double stranded or a single stranded, right. So, example, then gradually heated UV spectrometer say from 20 to 80 with 1 degree per minute, the UV absorbance is continuously recorded, and that we can get it the precise rise in the UV absorbance due to this hyperchromic shift. So, melting temperature we can find it out, right, melting temperature, and that is we can determine the rate of change of absorbance with temperature, and you can take the first derivative. So, the where it is maximal, that is basically 50% of DNA is in double stranded, 50% is the single stranded. So, that means alpha at that temperature is half, because half of the portion is in the double stranded, half of the portion is in single stranded, right. So, comparison of melting curve at different initial concentration demonstrate that Tm increases slightly that is what slightly it increases upon DNA concentration, not too much. If the DNA contents are more or less same, the Tm only shifts slightly, right. So, reflecting that improved duplex stability, if say 10 bases versus 15 bases versus 20 bases, if bases are more, the stability is slightly more. So, if that reflects that improved duplex stability at higher strand availability, if there is more number of nucleotides involved in duplex formation, the Tm goes up little bit. And now you can use all these parameters that we have determined to get the thermodynamic parameter, right. So, here a typical delta G RT ln K, right. This is thermodynamic equation, and K you can find the equilibrium constant. So, you can combine this to give the Van 't Hoff equation by eliminating the delta G, that is change in the Gibbs free energy of reaction. So, some jugglery of the equation. So, you can write this delta K equal to delta H by RT just from here, right, just divide by here and put it, you remember, let me just, delta G is delta H minus T delta S, and this is delta G equal to RT ln K, right. So, you combine this equation and you can find it out by arrangement this equation, right. So, from there you can rearrange to find 1 by delta T R by delta H ln absorbance by 2 and delta S by delta H. Now, if you look at this is more or less or you can find it, just rearrange this one and you can find this is a linear equation, and you can fit this linear equation delta absorbance by whatever concentration and here Tm, and it is a linear equation, you can find it out. So, this graph plots basically 1 by Tm against the ln of absorbance by 2. Therefore, a gradient of r by delta h, an intercept of delta s by delta r, just going back here, right, this equation. You plot this versus this is your intercept, this is your slope. So, you can find it out. That neglects the temperature dependence of delta S delta R. Reciprocal recorded of Tm plotted against the ln like absorbance ln A by 2, that is a linear regression determines the entropy, enthalpy and finally the stability, thermal stability, and that you can plot it in a Van duplex formation. So, if there is a CT mismatch, you can find it out that the stability changes and you can see clearly in the mismatch. So, by just putting it with a concentration dependent denaturation and measuring all the thermodynamic parameter, you can even find the mismatch that may be happening in the DNA duplex, right. So, essentially UV melting can be used for calorimetric measurement of DNA duplex stability, right. So, where we are going to use basically more commonly used due to accessible instrument, because UV visible is a relatively less costly instrument and UV melting can be used. Now, you can use same thing using differential scattering calorimetry, but that is comparatively expensive instrument. So, classical method, but less frequently used in calorimetry. Here you can get the UV melting just by UV visible. Instrument require like a UV melting can be used just UV spectrophotometer, temperature controlled sample chamber. Here you require calorimetry, differential scaling calorimetry to get the Tm and delta H and delta Cp, that you can get it from here. Here data collected is extremely simple, just UV absorbance versus temperature and determine the Tm. Now, you measure the heat flow associated with DNA melting or duplex formation. When it is melting or it is coming back, you can measure in calorimetry. Now, this is indirect estimation of thermodynamic parameter, that as we plotted here, we plotted absorbance and Tm, it is indirect. In calorimetry, you can directly measure all the parameter, right, direct measurement of thermodynamic parameter. One can get the delta H, delta S, delta G and Van 't Hoff analysis one can do, and in this case you can directly measure the delta H, then Cp and Tm, all those, right. It is a simple and convenient to perform rapid data acquisition. DSC requires some kind of like expertise. So, its experiment is insensitive and demanding, you cannot just, it is not as simple as UV visible. And then this is used for like a stability of DNA or PCR optimization. This is for detailed thermodynamic profiling or understanding the stability of DNA in research study, right. So, now since we have seen that Tm is an important parameter, right, it determines the duplex stability. Where we can use it, what are the factors that determines the Tm, what are the solution conditions that determines it. So, this is the equation that we had seen, this and this in a linear way, intercept and slope, right. So, one can get it and you can find it out the salt concentration actually that affects, right. You can put it that and you can find it out what are the basically parameters. So, here number of bases that are present and applies to DNA containing whether it is Watson Crick 80 or GC, and you can mismatch if it is happening. If you remember, we are here, we have seen the mismatch can be detected. So, similarly you can do all those analysis and in different salt concentration. So, mismatch containing duplex you can find it out. Right. So, parameters that dictates the DNA melting. So, the main important is what is the solvent condition, right. If solvent has something like formamide or urea, these are known denaturant, right. So, known denaturant of course will lower the Tm. How they are doing? Because formamide or urea, this basically disrupt the base pairing, disrupt the hydrogen bond, and actually this used for control stringency in hybridization, or in gel electrophoresis we use the urea denaturation, right. And the DMSO, it is organic solvent. of course, it lowers the DNA for most of these cases. And how it does it? Reduce the hydrophobic interaction, weakens the base stacking. You remember hydrogen bond and base stacking are two dominant stabilizing factors. So, this is useful in GC rich PCR template. If it is not melding, you just add DMSO, the TM will be lowered. Alcohol, something like ethanol isopropyl, that also lowers the TM. How it does? By changing the dielectric constant of the solution, and you can, like, this can be used for partial melting at lower temperature. Now, some agent like intercalating agent. So, intercalating agent, what they go and intercalate in the DNA bases, like ETBr, right, ethidium bromide, it raises the TM. So, what it does, inserts between the base pair and stabilizes duplex. So, if it is stabilizing, then base stacking is increasing, and due to that temperature TM raises. So, this is like a use for visualizing the DNA, or in topological study we can use it. Now, group binding ligands like proteins or other ligands, it can either decrease or increase depending upon what is the binding mode, and many regulatory protein use this mechanism, right. So, they can unwind the DNA topoisomerase and many other they are there that binds and can change the DNA stability. What are the other factors? These ions like you know negatively charged DNA. So, positive charge monovalent ion can increase the TM. If it is divalent ion, strongly increases the DNA. So, many chromatin condensing experiment you have these metals ion, right. So, this basically neutralize the phosphate charge, the negative charge on phosphate, and this more effectively you neutralize it, and because of that TM will increase, right. So, this is basically we have a common in a standard PCR experiment, and magnesium is essential for polymerase activity. Now, low salt concentration, it can decrease the TM. So, what happens that backbone repulsion that was there can be stabilized, and therefore, backbone repulsion increases and therefore destabilize the helix. Now, high ionic strength increases the TM. Chaotropic salts like lithium salts basically lowers the TM. So, these are the salt that can change the TM depending upon whether they are stabilizing or destabilizing. Similarly, pH plays important role. So, neutral pH, stable TM, it is for optimal base pairing or stacking. This is standard physiological condition, but if you lower the pH, lowering pH means more number of hydrogen ion added, that basically decreases the TM. So, what happens that bases gets protonated and they can, like, a loss of hydrogen bonding can happen and the helix gets destabilized. So, acidic condition DNA melts, high pH decreases again the TM, right, deprotonation happens and that disrupts the hydrogen bond. So, low pH is bad, high pH is also bad. So, many times in experiment you do alkaline treatment to denature DNA. You want to get rid of DNA, just change the pH of solution and then we can do. And moderately alkaline, this is like a slight decreaser or stable. So, using denaturation of plasmid, you just increase the pH and you can denature the plasmid, right. So, that is what one can do. So, where are we going to use this concept? So, you remember the quantitative PCR, right, qPCR, polymerase chain reaction. You want to know the DNA content of anything, then you, like any organism that you want to detect it, right. In detection device, all of us were knowing that in detection the PCR reaction were very deterministic tool for COVID detection, right. So, the nucleic acid can be quantitatively determined using PCR reaction. All the rapid antigen tests were fast, but the pathology lab we are giving a recommendation, if you have more than this or this color is appearing, you are COVID positive, but get tested with PCR. If you are seeing negative, but you cannot be so sure, get tested with PCR. So, this was a gold standard technique for detecting. So, you can use this for mutation detection or a diagnostic tool. The melt curve analysis in qPCR, as we see, TM gives the idea about the melting curve, and most common post PCR analytical method used for fluorescent dye like a cyber green that binds to only double stranded. So, you just put it double stranded, see how cyber green is getting used. So, procedure after amplification, the temperature is slowly raised, causing dye to dissociate as DNA gets denatured, and that decreases the fluorescence. So, fluorescence based experiment or sensor, you can make it by just using the cyber green, and as it amplify, temperature raising, the fluorescence decreases. That results into, you can plot the fluorescence that gives the melting curve. A single sharp peaks on the derivative curves to indicate that single specific PCR product has been formed. Cyber green is used in PCR reaction. Now, you can detect sequence differences, right. So, even a single pair base or mutation, single point mutation that is happening alters the stability of DNA duplex that results in a different TM, and you can detect it if the mutation is happening. So, if variants are coming for any pathogen, you can find it out just by melting curve. And then high resolution melting analysis is a very sensitive technique to differentiate the similar kind of DNA molecule or identify any mutation if there is a distinct melting profile. And finally, the homozygous versus heterozygous. So, this HRM method, high resolution melting, distinguish between the homozygous or heterozygous sequences, right. So, what we can summarize now, today we discussed or re-emphasized hypochromicity, low absorbance of double stranded DNA, hyperchromicity, high absorbance of single stranded DNA. And this transition provides a powerful tool for understanding the DNA stability, and we can use this concept for many of things. One of them is like a qPCR or HRM methods. This is used for research, diagnostic or many other molecular biology approach, right. So, you can make a device out of this to detect even a virus or a bacteria or any pathogen or mutation that is happening, right. So, this a simple, extremely simple technique has a multiple application, and only based on concept one can come up with a sensitive device for detecting any microbial infection that can be happening. Just you need a careful experiment design and a simple instrument that can detect the melting curve of a DNA. So, thank you very much and look forward to have you in the next class. Thank you.